You walked into a room and forgot why you went there. Your doctor said it's normal. It isn't. What's actually happening isn't just aging, it's a slow-burning inflammation that has been quietly activating for years. You stayed active, you watched what you eat, the fire kept burning because no one told you it was there. In April 2026, a team at Texas A&M published in the Journal of Extracellular Vesicles mice aged 18 months, the physiological equivalent of a 60-year-old human, 2 doses of a nasal spray, inflammatory markers in the brain reversed, memory improved measurably in weeks. The NIH funded it. In this video, I'm going to show you the exact mechanism behind that fire, why the nasal route changes everything, and what you can do right now while the science catches up.
That cross-section of a brain you just saw, the amber glow around the hippocampus, it isn't artistic. That's what activated inflammation looks like under a microscope. And the Texas A&M team published in the Journal of Extracellular Vesicles in April 2026 that this inflammatory state in animals physiologically equivalent to a 60-year-old was partially reversed with 2 intranasal doses of stem cell-derived particles. 2 doses, not a year of treatment, not a protocol. I'm not your doctor. And honestly, that's exactly why I can tell you what your doctor may not have had time to explain. My name is James Reyes. I'm an independent health researcher. I translate peer-reviewed evidence for adults who don't have time to read 300 pages of supplementary data. I got into this work through a hospital. My father had multiple strokes over a decade. I sat in more waiting rooms than I can count, and each time I watched the system focus on the episode, not the pattern, not the chronic, low-grade process underneath that was driving everything. I started reading the research myself because nobody else was connecting the dots for us. What I found when I got to this particular field, the neurobiology of brain aging, is that we've been asking the wrong question for decades. The question everyone asks is, why does memory decline with age? The question nobody answers is, what is actually happening at the cellular level? And is it reversible? 20 years ago, the dominant model was simple. Brain aging equals neuron loss. Neurons die, you lose function, linear, inevitable, pharmacologically irreversible. That model drove $1 trillion of drug development. Most of it failed. Today, the research tells a different story. The neurons aren't the first domino. The immune cells are. Nothing I say in this video is medical advice. What it is the research your doctor may not have read yet, because this particular finding is 6 weeks old. Drop a comment right now with just your age, nothing else. I want to see who's watching, because what I'm about to explain depends on exactly where you are. Here's what's actually happening inside your brain right now. Your brain has its own immune system. It's run by cells called microglia, and they are extraordinary when they work correctly. In a healthy young brain, microglia behave like maintenance workers, patrolling the tissue, clearing out cellular debris, protecting neurons, and going home when the job is done. But in the aging brain, and we're talking past 50, certainly past 60, something shifts. The microglia stop going home. They enter a state of chronic activation, not responding to a specific threat, just on permanently, alert. And in that state, they start releasing inflammatory compounds into the tissue they were supposed to protect. Researchers have a name for this, neuroinflammaging. And here's where, let me make sure I say this right, here's where the frame of normal aging breaks down completely. Because neuroinflammaging isn't a side effect of time. It isn't programmed into your DNA like gray hair. It's driven by specific signaling pathways, specific proteins, specific molecular switches that, in theory, can be turned down. Think about a woman who spent her 60s being told her memory issues were just age. Sharp in her 40s, sharp in her 50s. Then somewhere around 62, 63, she starts losing threads. Conversations where she forgets who said what. A name she's known for 30 years that takes 2 days to come back.
Her doctor ran the standard panel, all fine. Nobody checked her microglia. Nobody ever checks the microglia. That gap between what the research now shows about the cellular drivers of cognitive decline and what happens in a standard annual exam is not a conspiracy, it's a lag. The science moved faster than the clinical guidelines. But the lag has a cost. Every year it persists is a year the inflammation compounds. Has your doctor ever mentioned brain inflammation, or did the appointment focus on the same standard numbers it always does? The most dangerous thing about neuroinflammaging is that it accelerates in silence. No pain, no fever, no obvious signal, just a slow, persistent narrowing of the margins until the margins disappear. In the next block, I'm going to show you the two specific pathways the Texas A&M team targeted, because understanding them changes how you think about everything from sleep to diet to what questions to bring to your next appointment. The Texas A&M team wasn't working randomly.
They went after two specific targets, two molecular pathways that had already been implicated in neuroinflammation by a growing body of literature. The first is called the NLP3 inflammasome. I know that sounds like something out of a chemistry textbook. Give me 60 seconds to make it concrete. Inside a microglial cell, NLP3 functions like a fire alarm. When it's triggered by cellular debris, by viral fragments, by metabolic stress, it activates a cascade that floods the surrounding tissue with inflammatory signals. In a young immune system, this is tightly regulated. Alarm fires, response deploys, alarm shuts off. In an aging brain, the shutoff mechanism weakens. The alarm keeps ringing, the fire response stays deployed even when there's no fire. A 2021 review published in Nature Aging tracked the NLP3 pathway specifically in aged hippocampal tissue. The findings were consistent. As animals aged, NLP3 activation increased markedly, and it correlated directly with measurable cognitive decline. Not loosely associated, correlated. The second pathway is called cGAS-STING. Where NLP3 responds to environmental threats, cGAS-STING is designed to detect mitochondrial damage. As cells age, their mitochondria begin to leak. That leaked DNA triggers cGAS, which activates STING, which signals the immune system to respond as if the body is under attack. It isn't under attack, it's just old. Here's where, and I keep coming back to this because it's the piece most coverage missed, here's where the Texas A&M approach is structurally different. Most drug development in this space tried to block NLP3 or STING directly. The problem is that you need these pathways. Block them completely and you disrupt immune function across the board. What Shetty's team used instead were extracellular vesicles derived from human neural stem cells. These are biological packages, nanoparticles that cells naturally produce to communicate with each other. They carry microRNAs, short sequences of genetic material that act as volume controls for gene expression, not on-off switches, volume controls. The case for this approach, the EVs don't block the pathways, they modulate them. In the mice, this translated to reduced inflammatory activity in hippocampal microglia. The cells stopped shouting and returned closer to their normal surveillance state. The case against, or what the research also shows, is that translation from murine models to human neurobiology is notoriously difficult. The microRNA cargo that works in one model doesn't always transfer, and EVs produced at clinical grade have not yet been demonstrated at scale. In my view, the evidence leans toward this being a genuinely novel mechanism, not because it's new in the literature, but because it's the first time this class of biological vehicles has demonstrated this specific outcome in a hippocampal aging model. That matters. If this matches what you've been feeling but couldn't quite name, type yes in the comments. Think about a man in his late 60s whose father had dementia.
He's been watching himself for 20 years. He passed every cognitive screen his doctor ordered. He does the crossword, he exercises, he takes omega-3, but he can't shake the sense that something's happening underneath, a subtle slowing that none of his test results capture. A name here, a date there, the kind of thing that disappears by the time he's in the exam room. He isn't imagining it. What he's detecting is a pre-symptomatic phase that standard clinical tools weren't designed to measure. NLP3 inflammasome activation doesn't show up on a standard blood panel. Neither does cGAS-STING signaling. The tools your doctor uses to evaluate cognitive health, the Mini Mental State Examination, or the MoCA, are designed to detect deficits that have already occurred. They are not surveillance tools for what's coming. Here's the implication of that. By the time a standard cognitive screen flags a decline, the neuroinflammatory process that drove it has been running for years. The symptom is downstream. The fire started long before anyone noticed the smoke. That is the gap the Texas A&M research is trying to close, not by treating the symptom, by targeting the mechanism. And the mechanism, NLP3 and cGAS-STING hyperactivation in aging microglia, is for the first time a confirmed target with a confirmed intervention that worked in animals, under controlled conditions, with a delivery method that actually reached the hippocampus without surgery. That's not small. Nobody is saying it is ready, but nobody should be saying it doesn't matter either.
Before I get to the part that changes everything about the delivery mechanism, there's something about the blood-brain barrier you need to understand. Does this match what you've experienced, or were you told something completely different at your last appointment? Write this down. The blood-brain barrier is not a wall, it's a border crossing. And for decades, the entire field of neurological drug development has been trying to build vehicles that can cross it. Most failed, not because the drug didn't work, but because they couldn't get in. Here's the counterintuitive part. The answer was the nose. Not metaphorically, literally. The olfactory mucosa, the tissue lining the upper nasal cavity, is one of the few places in the human body where the nervous system is directly exposed to the environment. The olfactory nerve runs from the nasal cavity directly into the brain. No checkpoint, no barrier crossing required. When Shetty's team delivered the EVs intranasally, the particles were absorbed by the olfactory epithelium and transported along the olfactory nerve pathway directly into the hippocampus. The hippocampus, the structure most associated with memory consolidation, the area most affected by the neuroinflammatory cascade they were targeting. 2 doses, and the effect was measurable in weeks and persistent over months. Here's the counterintuitive part, and I want to be precise. The assumption is that treating brain inflammation requires chronic dosing, daily pill, lifelong maintenance. What this research suggests, and I'm being careful here because it's a preclinical finding, is that targeted modulation of the microglial transcriptome may not require chronic dosing. The EVs appear to reset the signaling state rather than suppress it. If that translates to humans, it changes the entire therapeutic model. Nobody told you the wall had a door. I pulled the full supplementary data 2 weeks ago. They sequenced the microglial transcriptome, not just measured a few inflammatory markers. 18,000 genes. The treated group showed significant downregulation of pro-inflammatory pathways and restoration of mitochondrial function. But here's the honest part. The mice were immunocompromised in the model. The cognitive improvements were partial, not reversal, reduction. The aging brain wasn't restored to 30 years old. The inflammation was brought down from a high baseline to something closer to normal. That's still extraordinary. Partial reduction of chronic neuroinflammation in an aging hippocampus without surgery, without systemic immunosuppression, through the nose, is a proof of concept that changes how researchers design the next phase of work. Here's the larger context that most coverage missed entirely.
Extracellular vesicles as therapeutic vehicles have been studied for over 15 years. The field spent most of that time trying to understand what EVs carry, their cargo, and whether that cargo could be engineered or selected for specific outcomes. The answer, increasingly, is yes. What makes Shetty's team's work distinctive is the combination of three things that had not been achieved together before: human-derived cell sources, a specific aging target in the hippocampus, and a non-invasive delivery route. Each of those individually had precedence. Together, in a preclinical aging model that yielded cognitive outcomes, this is the first published demonstration. And here's the thing about proofs of concept: they change the funding landscape. Research that produces a confirmed mechanism in a credible model receives attention and resources that theoretical work does not. The NIH funded this. The patent has been filed. The scientific community now has a target that works in an animal model, a delivery system that bypasses the blood-brain barrier, and a biological mechanism that doesn't require permanent pharmacological suppression. That's the starting line, not the finish line. Drop a number in the comments. Just your age. The patent has been filed. Dr. Shetty's team is working on producing EVs at clinical grade for human trials. That process takes years, not because the biology is uncertain, but because safety and regulatory requirements exist for good reasons. In my view, the most important takeaway isn't the spray, it's the target, because the pathways it addresses are modifiable by non-pharmaceutical means as well, and that's what I'm going to show you in the final block. The spray isn't available to you today. Human trials haven't started.
I want to be completely honest about that, because this channel doesn't exist to give you false hope about something that may still be years away. What is available to you today is the knowledge of what the targets are, and that knowledge changes what you do. Here are three things the research supports, with mechanisms attached, not vague lifestyle advice, but specific actions with specific pathways behind them. 1. Sleep. Non-negotiable. Microglial clearance of the brain, the process by which these immune cells clear out toxic debris and reset from their activated state, is almost entirely dependent on sleep, specifically on slow-wave sleep, which is the deep restorative stage that adults over 60 lose disproportionately. A 2019 study published in Science tracked glymphatic clearance in aged mice and found that sleep deprivation accelerated NLP3 activation in hippocampal tissue. The correlation held in human observational data. Adults with chronic short sleep, under 6.5 hours, showed higher inflammatory biomarkers, including IL-1B, which NLP3 directly produces. Your sleep is not a preference. It's the maintenance window for your brain's immune system. If you're waking up at 3 a.m. and can't go back, that deserves a clinical conversation, not just melatonin, a real conversation. 2. Mitochondrial support. The cGAS-STING pathway responds to mitochondrial DNA leakage, which increases with age as mitochondrial integrity declines. Zone 2 cardio, a pace where you can still hold a conversation, performed for 30 to 45 minutes 3 to 4 times per week, has been shown in multiple independent studies to improve mitochondrial biogenesis and reduce markers of mitochondrial stress in aged tissue. The effect appears within 8 to 12 weeks of consistent practice. Think of a 67-year-old woman who started walking with a neighbor, 40 minutes, 3 mornings a week, a pace where they could still catch up on the neighborhood news. At her 6-month check, she mentioned, almost casually, that she felt sharper. Her doctor attributed it to social engagement. The mitochondria had something to do with it too. 3. The conversation to have. Walk into your next appointment with a specific question. Not, how's my memory doing? Not, is this normal? This. Have you seen the 2026 Texas A&M study on intranasal extracellular vesicles? And is there anything in my current profile that suggests elevated neuroinflammation worth tracking? You may get a blank look. That's information. The study is 6 weeks old. Your doctor may not have had time to read it, but by asking the question, you've changed the nature of the appointment. You are not a passive recipient of medical decisions. You are an informed participant in your own health. Take these 3 steps to your next appointment, not instead of your doctor, alongside them. That's how this works. Let me close the loop I opened in Block 1. 3 things to walk away with. 1. Your brain has a chronic inflammatory state, neuroinflammaging, driven by 2 specific pathways, NLP3 and cGAS-STING. 2. In April 2026, a Texas A&M team showed that extracellular vesicles delivered intranasally can modulate these pathways in an animal model equivalent to a 60-year-old human. 2 doses, measurable improvement in weeks. 3. The spray isn't available yet, but the targets are sleep, mitochondrial maintenance, and an informed conversation with your doctor. If you're walking away from this video differently than you walked in, drop "informed" in the comments. If this is the kind of research you want to keep seeing, the subscribe button is right there. I don't post every day. When I post, it matters. There's someone in your life who should hear this before their next appointment. You know who it is. Share this with them. Of the 3 steps I just gave you, sleep, mitochondrial work, and the doctor question, which one are you starting this week? Tell me in the comments. I read the research, so you can walk into that office and have a real conversation, not just nod and leave with a prescription you didn't fully understand. Nothing in this video is medical advice. James Reyes is an independent researcher, not a clinician. The findings I've described come from a preclinical study, animal model, not human trial. Consult your physician before making any changes to your health routine.